Good morning to all of you. On behalf of Sri Lanka College of Internal Medicine, I wish to welcome all of you joining physically as well as virtually. This is a hybrid workshop. We got around 150 people are registered for virtual participation. So as a college, we have given a lot of emphasis on developing research culture among our members, and we have been conducting series of lectures and workshops and one-to-one research guidance during last couple of years. I think our effort has been paid off, as there is more research done by our members. Even though we did a lot of things on research, we couldn't do we couldn't do a workshop on manuscript writing. So this is going to be the first manuscript writing workshop done by our college. Translating what you did in your research into a manuscript is very, very important. That is the only way. A good manuscript is the only way that you can sell your research work as a publication. So therefore, writing a good manuscript is very, very important. I'm sure today we are going to learn how to write good manuscript, because we got 3 eminent speakers of top caliber. So they are going to teach us how to write a good manuscript. I just want to say a few words about research guide also. Our college has been conducting one-to-one research guide to help the trainees to conduct their research, and we have been doing this for last 2 years. And in future also we are planning to do this. If there is anybody who is interested to get some help, please let the college know. We are happy to help you. It doesn't really matter whether you are doing medicine or whether other paraclinical or clinical subjects. We are still happy to help you. So if there is anybody who is interested in doing research and wanted to get some help, please let us know. So we are happy to sort of help whatever the possible way to improve your research activity. We got in this workshop, we are going to cover all the areas: title, abstract, introduction, and then of course methodology and the result section, and also the lastly discussion and conclusion and reference. As I said earlier, we got 3 eminent speakers of a caliber of Professor Saroj Jayasinghe, Professor Sarath Lekamwasam, and Professor Anuja Premawardana, and all of them are top-class clinical researchers in Sri Lanka. So I'm sure you will be able to learn a lot from this workshop. To start the workshop, now I would like to invite the President of Sri Lanka College of Internal Medicine, Dr. Kumudini Jayasinghe. She will join us virtually. So over to you, Kumudini.
Thank you, Professor Herath. Good morning, everyone. I warmly welcome all of you to this first workshop on manuscript writing organized by our college, Sri Lanka College of Internal Medicine. As Professor Herath said, publishing a research paper is a huge accomplishment for a researcher. And to be successful in publishing a research paper, it must be well written. To help our young researchers on this task of manuscript writing, SLCIM has organized this workshop today. And we have invited 3 experts as resource persons who has been already introduced to the audience today. And they have done a tremendous work in research. And I must thank Professor Saroj Jayasinghe, Professor Sarath Lekamwasam, and Professor Anuja Premawardana for their contribution as resource persons today. And I must thank Professor Herath, HMM Herath, the leader of the SLCIM research wing, for organizing this workshop as well as other research activities of the college. And while thanking the organizing committee, I invite all of you to get the maximum benefit from today's program. I'm sure this workshop will help you to write your research paper confidently. Have a fruitful workshop. Thank you very much.
To talk to us how to write title, abstract, keywords, and introduction, may I introduce Professor Saroj Jayasinghe. And Professor Saroj Jayasinghe need no introduction. All of us know who he is. He's the Emeritus Professor of Medicine, University of Colombo. Can I invite you, sir, please?
So thank you very much once again for inviting me to make this short presentation. Since it's a workshop, I think we must try our best to be interactive. So may I suggest those who are here as well as those who are on Zoom to take out a piece of paper and think of a project you want to write up. It could be something which you have done or something which you imagine you have done, and try to work out the sort of processes things we are going to tell you or discuss. So my topic is, it should be taken as a cluster because that's the area of a paper which makes the reader want to read more. So analytics have shown that it's the title. In fact, if you look at Google Analytics is always watching what you are doing with your search, and they find that in most instances we look at the title of a search, medical or otherwise, then the first few sentences. If that's interesting, then you go to the other sections. So the largest group of readers will look at your title. Then a smaller number will look at the abstract. And then still a smaller number will look at the introduction, and only a fraction will read the whole article. So that's a fact. And we have to play around with this audience. So the importance of the title is highlighted because of this, because if your title is attractive, then the people will get on to the next stage. Keywords is something which you use to optimize search. You might find the article based on the keyword, but your interest to read depends on the title and abstract. The keyword helps readers to get to your article. So this cluster is important if you want to share your work. So when we talk of a title, now as part of the interactive thing, I want you to write your own titles. And no one is going to ask you questions. You'll be self-evaluating what you write as you go along. But think of a title. If you can write it down, because even during ward rounds, I used to write down the diagnosis. Then you are committed. You're sticking out your neck. Right? So this is on your own. You can always write it down. Write down the title of a paper you want to submit to a journal. Now, the title also maximizes your search. Keywords are things which you put in, you log in, when you log into a database, you write certain words, and from the database you will find the articles. But similarly, the title also helps you to attract readers to your paper. So what we call maximizing searchability. And if your title is attractive, then it will engage your readers. So it has to give a significant amount of information which will engage your readers. Okay. Now, in addition to that, there are few tricks or few tips which we mentioned about the title. This is all from literature, which is available, by the way. All these big publishing houses like PLOS or Springer Nature or Elsevier, they have guides for authors, for editors, and so on. There are super videos. Actually, once you access those, you will find that we are redundant because it's all there. And they have really got the expert editors, expert writers to describe how you write, how you edit, how you peer review, and the whole process. So it's there. I'll give you some of that reference, and it's given here. That's the PLOS, which talks about the title. Now, in your title, you try to include some of the keywords, because your keywords are key. So if your keywords are key, there should be important in your title. And you must include the most important information about what the target audience will want, and also the design. Some, whether it's a clinical trial, a descriptive study, and so on. And also it must bring out something which you've discovered or tried to discover. So now you've written down your title. See whether you have, the first one you wouldn't have still worked out the keywords, but does it bring out to your audience something which will make them interested to read on? Has it brought out something about your design? Is it a case series or whatever? And has it given a hint as to what you have discovered? Now, there are a few do's and don'ts. So you have to keep it concise and informative. You write for your audience. You have to entice the readers. You incorporate the keywords. And you write it in sentence case. Now, whether you want to have capitals with each word, the journals will tell you, but if you write it in a usual sentence case, that's okay. That's starting with the capitals and capitals when otherwise when needed. Now, when you say concise, some journals will give you the amount of letters or characters which can be used. But as a general ball mark, about 12, 13, 14 words would be ideal. Okay? And you have to aim for the audience. So if you're writing to a medical student's journal, what you're going to say would be different to what you would do if you're writing to a high-end specialist journal. The words you use will be different. And you try to incorporate some of the keywords, and as I mentioned, the capitalization. A few things not to do. Avoid writing your title as a question. Now, I used to do that quite frequently. But what they say is that when you try to bring out a question, it doesn't really. You bring out a question and very soon you know the answer, because in the abstract you give the answer. And then your interest about reading the whole paper goes off, right? And also don't sensationalize research. For certain magazines you might want to sensationalize. But otherwise you have to use the proper technical terms when you're writing about your research. Okay, now we'll go through a few examples. So in these examples, this is an example for a clinical trial. And you'll see that the key elements are being covered. The intervention, and in this instance, it's herbal preparation. about a disease and its symptomatology and where it was carried out, and something about the design. So using a herbal preparation, whatever, to reduce respiratory symptoms in COVID in Sri Lanka, a randomized clinical trial really gives you the summary of what you want to say. So if you are interested in herbal preparations and treatment of COVID, you will go on to read it. Effectiveness of treatment for a particular disease in a population and based on the outcome, a systematic review. So for a systematic review, again you are using the same structure, but you will see you have mentioned what the disease is, you have mentioned about the population, you have stated the intervention and the outcome, and also what type of study. Again, captures the whole thing. So for a clinical trial as well as for a systematic review, you have given the key elements and it's standalone. You understand what it's all about. So in the clinical characteristics, again, it's the same. You're giving something about the disease, what you're looking for, what the population is, and what the location is, and maybe even the type of study you want to do, descriptive study, prospective study, case series, or whatever. Okay? So now you have written your title. Try to improve on the titles you have written. You can show your neighbors and see what their comments are, and see if you can improve on the title you wrote for your submission. Okay, so next is we get on to the abstract. Now, abstract will vary from 150 to 300 words. Most places ask for 250, and most conferences are also 250 abstracts. So you have to get used to that. That is to be able to tell the whole study in a summary form in 250 words. That calls for a lot of discipline, and also you have to create readers' interest, and also use your language in a very efficient manner. So an abstract differs from the introduction. Okay? Yeah, abstract, you are trying to give an overview of the study. You are saying when you read the abstract, you will know what the study was, how it was done, what are the results, and what is the impact and its relevance. So that's why a lot of people read only the abstract, and that's not something good. If you want to comment on a paper, you must read the whole paper. The authors can play around with words and give limited information in the abstract, which might be slightly away from the true results or the true relevance. So the abstract differs from an introduction. Here you're going to, it's a standalone. Introduction is different because it's a broad introduction to the whole topic. It is trying to sell you to read the rest. The introduction is trying to sell you and convince you that you must try to read the rest. Whereas the abstract is a standalone summary of everything what you've done. So what is it? What is your paper about? Why is it important? How did you do it? What did you find? And what are your findings? Why are your findings important? Some journals want it classified according to introduction, objectives, methods, results, and discussion or conclusion. Some don't. I personally think the journals should And certainly some of these conferences should have it fairly flexible, because there are certain studies which will not fit into this straitjacket, this compartmentalized way of looking at it, especially qualitative studies. So if you try to remember what you want to include in an abstract, it broadly follows the IMRAD. You have heard of IMRAD, Introduction, Methods, Results, Discussion. That's what we usually structure the whole paper. So abstract also is somewhat similar and might give a bit more on the implications or the importance of the study. Okay, so if you look at this, you will see how the classification is. Right? And when you write an abstract, once you do this, right, you have to spell check. Use a free program like Grammarly, which will correct your grammar. And now we have ChatGPT. So this is coming in. Let's face it, you will be getting your abstracts written by artificial intelligence. Going to raise a lot of ethical issues, and still the discussions are going on as to how, whether it should be acknowledged, how it should be acknowledged, and so on. But you should use the software which is freely available to correct your grammar. And Grammarly is very useful because it gives a score. And the free program also allows you a lot of improvement in your paper. I hate grammar, so this is lovely because I use this very frequently. Right, keywords. So I've told you about the title, the abstract, and now as regards to keywords, there's a bit more of information, or what shall I say, more technique involved, because you're trying to optimize your keywords so that when you enter the keywords into the database, your paper will be flagged. If your paper is flagged, there will be more people reading. If more people read, it will be critiqued or cited. And that's what you want. You want the scientific community to read your paper, right? So it has to represent the main concepts and describe the topic. And you try to link it to the title. As I mentioned, you try to link all these to as much as possible, the title, keywords, and abstract, because each one complements the other. So I'll leave it to you to read. So you try to think of the audience, think of yourself, or you can ask a few people even, and select the keywords which will capture your article, which are likely to be picked up when you do a search. And the keywords must be linked to the abstract as well as the title. Now, how are you going to search or identify these keywords? Some of the databases will give you a list of terms. The medical subheadings, MESH is one such system from PubMed. And if you use the terms which are there in PubMed as your keywords, then you are likely to have a larger readership. Okay? So if your title and abstract also has those keywords which are used in MESH, you are likely to be detected when they searched for articles, because people will be using the mesh terms to search. And now mesh is, you can do mesh on demand. Now certainly I'm not an expert in this area, but I've used this, and that is where if the librarians are, this is a library science sort of thing, but you just add your text and it will give you a few keywords. But there can be a few problems in this, because I feel you have to use your own creativity depending on what exactly your paper is. And you realize that if your paper is going outside the usual knowledge domain, mesh might not capture it. I'll show you an example. Now, this is Professor Rath's paper. So I've taken the abstract and I put it into mesh on demand. And that's what mesh on demand gave, and that's what the keywords they have selected. Okay, so it's what you have to do is to use this, plus use your own judgment as to what keywords you want to use. I told you, sometimes you go out of your comfort zone. So this is a paper I've written. It's been submitted, Decolonizing Postgraduate Medicine, a case study of postgraduate training in Sri Lanka. Outside the box. And when I put that into mesh on demand, that's the, in fact, because I talk about MRCP, it's talking about cholangiopancreatography, MRCP. Whereas this is my list of keywords. So it all goes to show that you don't believe the machine all the time. You can use it and use a mix of words which are there from mesh and what you think is important. Finally, we come to introduction. How do you write an introduction? And a good introduction gets the golden passageway to the study, because if it is a good introduction, it will sell you the article for people to read. So you give the known, then you give the unknown, and then you say, develop a hypothesis or a research question and tell what you're going to do. In this, you can have the objectives also, right? So you have to give top-class references for what is known. Good references, high impact. And then you argue and say, this is the part which is not known, and therefore this is how I'm going to explain it, and roughly the methodology. So these are simple guides, right? 10 to 15% of the total length, simple sentences in the present sense, present tense, so that it's current. And you may have to use abbreviations. That's the time you introduce abbreviations like MRCP or whatever, right? And you have to have good references. Please don't start with, when you talk of leptospirosis, how leptospirosis was discovered and that it was an organism which was found by so and so in such and such a center. That is not required now. You're writing to experts, you're writing to your colleagues, your peers. That's a magazine where you are writing it to medical students. You want to talk about leptospirosis and how it was discovered. Okay? Unless, of course, the paper is on history of leptospirosis. And the references in this section should be to papers published in quality journals. This is an introduction from the previous era, right? Neurotoxic effects of organophosphorus insecticides an intermediate syndrome. Some of us have known these authors like Professor Nimal Senanayaka and Professor Lakshman Karliadda from Peradeniya. Let's read that introduction to a case series. Now try to figure out where they have mentioned in the introduction the known. Then they have mentioned about the unknown. So the known is acute neurotoxic effects during cholinergic phase of organophosphorus poisoning are well known and given a top reference. The pathophysiological process underlying this syndrome seems to be different from that of the cholinergic crisis and the delayed neuropathy. Unknown. Right? And the three clinical situations suggest a triphasic effect of organophosphorus compounds after human intoxication, like a hypothesis. Okay? This paper describes an intermediate syndrome of neurotoxic effects that appear after the acute cholinergic crisis, but before the expected onset of delayed neuropathy. That's the method. Right? So your introduction has really given the interest to the reader. You want to read more. Right, so in summary, what I've done is, the Y has fallen for some reason, but you see, if you really look at it, all these are linked. The title, abstract, introduction, these are linked. The keywords are the things which will help to find your paper. If you plan it in such a way, you will be able to look at, you should look at this as a cluster and plan your paper in such a way. Now, here is a book, right? Can't read it from here. How to practice academic medicine and publish from developing countries, right? That's from Springer. That's free. You can download the book. And it will give you most of the things which we've discussed. Thank you. Thank you very much.
Thank you, sir. Thank you for the excellent lecture. The next lecture is on methodology and the research section, which I believe is the most important part of the manuscript, and that is the engine of your manuscript. That has to be the most difficult part, I suppose. So we have the best person to talk to us on writing the methodology and the research section. It is none other than Professor Sarath Lekam. He's from the same faculty where I'm also coming from. He's from the Faculty of Medicine, University of Ruhuna. And Professor Lekam is one of the top rank researcher in the field of osteoporosis. So over you to sir.
So first of all, thank you, Herath, for the very kind introduction. And I'm just trying to figure out, I'm about to retire now. I don't know why people still sit in the back of. It's something I have not been able to figure out from my student days. Like I also did that. Okay, I'll try my best to tell you how to write this particular two section that Herath said most important part of the manuscript about the methods and the results. Professor Saroj touched this very briefly. That's about the Sir Bradford Hill's questions, and which are famous in medical writing. And in fact, Sir Bradford Hills is the one who is one of the English epidemiologists and who did quite a lot for the clinical studies, randomized clinical trials. And at a point, he was considered as a father of clinical trials. And he simply said that in the manuscript, what you're trying to say is that basically address what did you do, why did you do that, that's introduction, and what did you do, that's under methods, and that comes the results, what did you find, and then of course a final discussion, what does all it mean. But if you really need to get an idea about the writing methods, and then there are plenty of guidelines in the internet. And what is very well known, one of the very well known one is what is called the consort document. I think people who are involved with what is called randomized clinical trials, and then they know very well about the consort document, that describes what are the essential components in the clinical trial. And also when you're reporting a clinical trial in manuscript, what are the essential components. So likewise, there are what are called PRISMA, that is for the meta-analysis and the systematic reviews, and there are a few others as well, like STARD and the MOOS and other kind of documents. So if someone is really interested of writing a manuscript, and depending on the type of the research, should start reading one of these. Now, I will give a little background into what is the importance of the methods and the results section. Let's say a research found an association between, let's say, A and B. Now, there are a couple of possibilities. This could be complete due to bias that committed during the research. Okay? And bias is, for the people who are in the clinical field, bias is what's called systematic error that you have committed during the research. It's not a random error, it's a systematic error. Let's say it can be due to confounder. Confounder gives an alternative explanation for the association between A and B. For example, let's say if you find the coffee drinking and the association of coffee drinking with let's say, a link up with a heart attack, and somebody might say it's not actually coffee drinking, it's a smoking, because smoking and coffee drinking are linked, and the smoking and heart attacks are also linked. So it's actually not a direct relationship between coffee drinking and heart attack, it actually works through the maybe smoking. So it's a confounder. And it just could be due to a chance finding. And also it can be a real biological explanation behind it. Let's say how you tackle this. Now, once you said it's not due to bias, and also you eliminate the possibility that it's due to confounder, and also say it's not due to a chance, and then you convince the reader saying that this is a real association between A and B. It's a biologically meaningful association between A and B. To say that it is a biological meaningful association, you must say it is not a bias, it's not due to confounder, and also it's not due to chance. Now we do that. Now the biases and the confounders, bias is dealt in the methodology. So in the methodology you write in a manner saying that there was no chance for the bias to actually come into the methodology. So methodology did not include any bias. The confounders, partly methodology deal with the confounders, but then the results also, what is called statistical methods also can deal with the confounders. For example, let's say that there's a gender difference between let's say whatever the association between A and B, and then of course you can run different statistical analysis and say it is there among the men but not among the women. So that can be done. The possibility of the chance as an explanation association between A and B is eliminated by the statistics. So actually the role of the statistics is mainly to say it is not due to a chance. Now, once you have done that, and then of course you say there's association. Okay. Now, methods and the results are the two major parts in the manuscript, and they are linked. That's what I want you to understand. They are not actually the parts of the manuscript that just standalone. They are standalone, all right, but they are linked in some way. How they are linked is that the incorrect methods lead to what are called incorrect results and the conclusions. And then you get what are called inaccurate conclusions, and also the credibility of the findings become highly questionable. Now, there are technical reasons why some manuscripts are rejected. But there are what are called non-technical reasons. For example, if you send your article into a journal which does not address that particular area, like you send epidemiological study into a journal which is highly technical stuff, publishes on highly technical stuff, that will get rejected because it's not their kind of article that they look for. But there are what are called technical reasons why a manuscript is rejected. Most of those are in the area of what are called the methods and the results, right? Incomplete data, poor analysis, inappropriate methodology, and answering the hypothesis. So those are the ones that people say why manuscripts are generally rejected. Now, is there a way that once you have written your methodology and say check for the adequacy, whether you have written enough? Is there a check? And then if you have, let's say, once you have written and then read through it and then say, hang on, I have given sufficient information or the details that if someone else wants to repeat this study, then he can repeat it. So if you think that you have given enough information about the study that you had done so that somebody else can repeat the study in the same manner, then you have given adequate information in the methods. The methods had 3 key areas. There are what are called study subjects. So most of the studies, they have what are called study subjects. And then there are what are called interventions. There can be measurements or maybe observations. So you study subjects, you just don't let them be there. You do something. You at least, if you don't intervene, you make some observations about them. And then that information is analyzed. So those are the 3 main areas in the method section. The study subjects, why and how those people were included in the study. Also in that number, for example, why is that 100 people have been studied and not 200 people? So you need to explain why and how these people got into the study, and also in that number. They can be animals, lab specimens, or human, and the humans can be different, like cases, controls, exposed, non-exposed. So there can be various type of people in your study, and then you have to tell the reader why they are being selected. Now, methods, the interventions or the measurements or observation that you have made, you need to describe them operational terms. Operational terms means that how they were really measured. For example, let's say case definition of leptospirosis. How the case of leptospirosis was defined in your study. It would have maybe different. You have taken a different approach in defining leptospirosis in your study, but you must tell how the particular cases were defined in this in your study. And also the method section is written in the chronological order, like let's say selection of subjects, interventions or observation, and then the outcomes, definition and measurements, and the method of analysis. So finally you end up with a statistical description on a separate section on a statistical analysis. Like Professor Jayasinghe said that the introduction is written in the present tense, the actual method is written in the past tense. But generally, what we say is that introduction is written in the past tense. We find lots of people write the introduction in past tense, which is wrong, saying that, so and so found these things. It should not be actually written in the past tense, saying that, these are to improve the clarity of the sentence. What we find is most of the, sometime in the methodologies that are written in the future tense. That means we have that cut and copied and pasted that from the proposal, and without changing actually anything, they actually taken the cut and pasted the proposal into the method section. So that is in the future tense. Actually, they are not changed. So it should be written in the past tense because that you have completed that work. And also you need to maintain what is called internal consistency. The internal consistency and the method and the results section should be concordant. That means say that you should not describe a procedure if results are not going to be shown. Because it's useless really, because it's redundant. Then you describe something in detail, but the results are not going to be shown. And also that you should not display results for procedures that has not been described in the method. So that's why they say there should be internal consistency between the results and the method sections. study design. Also, you need to describe that, and the participants, who are the participants, like, who are the cases, case definition, and of course, the controls. When there are interventions, you need to tell the readers about the drugs and the doses, about the manufacturers, the placebo, whether they're matching, unmatching, and adherence to intervention, and degree of supervision. And then usually you supplement that with what's called the diagram when it comes to a randomized controlled trial. That is very well described in what's called in the consort. Outcome measures and how they were measured, what instruments were used for the measurements, like what's called precision and the accuracy of the instruments are generally, you require to say that you have used this particular instrument, and then the instrument has got accuracy and the precision of this much. So I'll come to that in what is accuracy and the precision. And if the questionnaire has been used and the validity of the questionnaire has got a validity that can be measured in various ways, and also linguistic adaptation, whether it has been adapted into a local language, and who measured, and people who are involved in the measuring, and also under what condition, like the temperature, and then how they were measured, fasting conditions, subjects came on out of overnight fasting, and the correct temperature has been maintained during the procedures, and all that need to be described. And the accuracy and the precision is something that clinicians need to understand when you come to certain procedures, a measurement procedure. Accuracy is that there is basically, let's say, if you have got a bull's eye, and whether you can hit the bullseye, the center of the bullseye, and whether you can hit the center, right in the center, and also whether you can do all the time if you are doing it repeatedly. You can see that number one here is hitting at the center. So this is accurate because it is hitting at the center, and it is precise because it is hitting the center all the time. Now this is not that inaccurate, because if you look at the distance from the center to the measurement is not very far. So that's very accurate. There's a little bit of accuracy error. So that you have to live with accuracy error. But then you can say it's not precise because it is in all over the place. Now this is not accurate because you can see that it is far from the center. So it's not accurate, but it's precise. Okay? And you can have a real situation. For example, let's say you go and ask a question from your boss and he gives a wrong answer. And you go and half an hour later go and ask a question and he gives a wrong answer. And once again he gives a wrong answer. You can say, my boss is not accurate, but at least he's precise. Because he gives a wrong answer all the time. So it can be that. And also now these are not accurate. They are neither accurate nor precise. So they are not hitting close to the center, and also it's hitting all over the place. So measurements have, when you come to biological measurements, and the biological measurements have these two key elements in the measurements called accuracy and the precision. And sometimes when you say you have used a certain measurement tool, and then reviewers might ask, what is the accuracy and the precision of your instrument? That's what they mean. The statistics indicate the tests that were used and define the p-value. Remember, it is not always p less than 0.05. Because 0.05 is all right if you are doing on a limited number of comparisons. Remember, if you are doing multiple comparisons, the p need to be corrected for the multiple comparison. Because you know that the p is set at with 1 in 20. A chance is given 1 in 20. But if you are doing a lot of comparisons in a lot of subgroup analysis and all, and p need to be corrected. But there are various ways of doing that. Famous ones are what's called Turkey and the Bonferroni. And Bonferroni and Turkey adjustments of the p-value are the common adjustments for the multiple comparisons. And some journals, they insist on the confidence intervals, or credible intervals or confidence intervals, and they believe in those more than p-values. So they might say that just give the confidence intervals around those estimates. The types of software and the version, and then remember there should be a legality, because you should have a licensed copy of that particular software. So remember, if you don't have a licensed version of that software, and it's a bit of risky thing, because you are under the law, because you had violated the law. So that become a bit of problem. Let's say content of the well-written method section. Does the text describe how the research question was addressed and how trustworthy are the measurements of the variable and the consideration? So this is what the reviewers go through. Were these measurements recorded, analyzed, and interpreted in the correct manner? Would the reader be able to repeat the experiment in the same way? So this tells the usual reader whether you have done a good job. What are the common problems in the methods? And do not adequately describe the cases and controls, so that the reader will find that there's a little overlap between the cases and the controls, right? So let's say cases are picked up, on the, let's say, acute coronary syndrome, and then you pick up the controls, age and the gender match people who have not had acute coronary syndrome. But then what guarantee you have that, this fellow who is taken as a control will not get a heart attack next month, right? Because he may have 90% block in his coronary and then waiting to get a heart attack. Maybe that, we had completed a study taking him as a control, but the next month he's going to get a heart attack. That might happen. So he's got 90% block. So there's an overlap between the case and control. The recruitment criteria, because you must avoid what's called selection bias. This is one of the bias that we generally see in the research. So selection bias should not be there. So people say that we are taken, let's say, convenience sample, collected people from Monday to Friday, not over the weekend, and again Monday to Friday, and also that's not correct. That's because you ignore a certain people who get admitted to the hospital during the weekend, and they can be completely different from the people who get admitted from Monday to Friday. And the method of randomization, whether you use a credible method of randomization like a block randomization or stratification, that type of thing. The degree of blinding and how they maintain if it is relevant for the study method. Quickly go through the results and then what was found. Now the results section is composed of, it contains what are called words, and words tells a story, and there are tables. Table summarizes the evidence. I will spend little time on the table, how to construct a good table. Illustration, they highlight the main findings in a graphical manner. The statistics support all the statements. So that's actually what the results section composed of. You need to organize the results in the natural order. Give the baseline or descriptive data of the entire study sample. So you first tell, this is what my study people look like. The mean age and the standard deviation. So people can figure out and see how the age sort of range in this study sample, and also gender, percentage of women and percentage of men. And then you get what are called the main results, which will answer your main objective, and then you go for what are called the post-hoc subgroup analysis, right? That you thought may be relevant, and then you try to see whether there's a gender difference, whether there's age, where there's a difference, subgroup analysis. And also must give it as uncluttered as possible, because people tend to give results in a very awkward manner, so no one can understand, So it should be brief and uncluttered. And also most appropriate way to illustrate the results, you have to think about it. Begin with the description of participants, and then that will tell you how representative they are with whatever the population you're going to apply those results. Okay? And if there's more than one group, compare them at the very beginning to tell what are the differences between these two groups and how these groups are comparable, like at the very beginning. And avoid the term demography, which I do not like in my manuscripts. Demography, just say descriptive data, which is more simple thing. Answer to your main question first, and then give positive and negative findings, and also give unexpected finding if meaningful. You found something which was not expected in your main study, but if it's clinically meaningful or clinically or biologically meaningful, you can show that, but otherwise you don't have to show that. Now, many people, many unable to differentiate what is between the data and the results. What is data? Data is actual findings like numbers, percentages, like measurements and all. And there are general row or summarized data, maybe mean value, the standard deviation, maybe transform percentage from the baseline. So they are called data. The results is little different because there should be a meaning of the data. Like men were heavier and taller compared to women. So that's actual results. So there's a little difference between the two. And the good, well-written results section should have actually a mixture of both. Now, what's the correct combination of the data and the results? Let's say 12 months in 14 untreated patients, the mean blood glucose concentration was 205, given a standard deviation. And in 16 people treated with the drug X, the mean blood glucose concentration was 105. So you can write that. But I don't know whether this data information has already been given in the table. If it is given in the table, don't write it, because you don't have duplicate. I'll come to that later. If I was writing that, I would write in this manner. I said 12 months, the mean blood glucose concentration was nearly 50% lower in 16 patients treated with drug X compared to 14 untreated patients. So I'm giving a little bit of results actually, but I'm giving the data as well. I give 205 standard deviation 12 versus 105 standard deviation 10 and give a p-value. So that will support my what's called the claim or statement that I made. So this is much nicer when you read that. And remember that people who are reading that are not 100% scientific people. There are clinicians, there are students, there are many people, and should be readable actually. And a paper in the manuscript say that you enter from the introduction, get out of a discussion, and there should be a journey through that. It should be you should facilitate that journey. Not making it too much technical really. And also, writing uncertainty bringing into the manuscript. For example, we were unable to demonstrate the existence of H. pylori organisms in the biopsy samples. So there was a bit of uncertainty. We were unable. Say no H. pylori organisms were found in the biopsy samples, so that's plain and simple thing. And do not overdo it. Don't say a marked or slight reduction of systolic blood pressure. Don't try to quantify it. And some people, they go on to say highly significant reduction of blood pressure. Put a p-value into that and they highly significant reduction of systolic blood pressure. And say very high correlation for when you see when you get excited about a R value of 0.8, like very high correlation. In fact, Mark Twain is a USA journalist, and a lot of his work, his statement can be taken to medical writing actually. And he's the one who said about the abstract. And he said writing abstract is so difficult actually than writing a manuscript. And he said, I had no time to write a short letter, so I sending a long letter. So because writing abstract is actually a little more complicated than that, because we have a luxury of writing words, so many words in the manuscript, but that you don't have in the abstract. So you don't have to think about words so much when you're writing a long letter. He said, substitute damn every time you are inclined to write very, and your editor, chief editor, will cut all of them off, and then it'll be fine. Right? So don't try to overdo it, like when you see the p-value less than 0.000. And that may be because that your sample size is too large, and then you got that p-value as a result of that. There can be another explanation for that p-value. But then don't try to overdo it in a manuscript. Okay? There can be other, more than biological association, there can be other reasons why you get that p-value. When you write in a p-value, give the exact value, and only where necessary. Never use NS, because some people write not significant NS. Because really there's no difference between, as far as I'm concerned, there's no difference between p-value of 0.051 and 0.049. For one, maybe one for the NS, for the non-significant, other one may be significant, but as far as I know that, there's hardly a difference between these two values. This is another problem that you have, but it's called you P all over the paper, right? Whenever you write something, you say there was no significant increase in systolic or diastolic blood pressure, you give a p-value, heart rate, give a p-value, respiratory rate, another p-value, and oxygen requirement p-value. So it's called peeing all over the manuscript, right? So don't do that, right? You can write all that and then give one p-value. Then uses and misuses statistics. So that's one of the main reasons for rejecting a paper, I told you earlier, and either badly chosen or badly presented, and you stick to general principles of statistics. The unnecessary precision is often find you in the paper manuscript. Like, body weight of women, 67.1. You don't have to give that precision. You can say very well 67.1. That's enough. You can leave that in your analytical column. When you are using your whatever the software that you are using for the analysis, you can leave it like that. That comes for the analysis, but not for the reporting. Yeah, some people say mean age of the subject is 45.52. Now, what is 45.52? I can't think of that. 45 is okay, 45.5 is okay, no problem. But 45.52, which I can't figure out. So why don't you leave it at 45.5? I mean, that's really fine, because the reader will understand it. Should not give reader something that he can't figure out. Exceptions can be, there are certain measurements that need go into decimals, like, for example, bone mineral density. Now that need to go to 3 decimals. So that you have to do, or T scores, Z scores need to go to 2 decimals. So that's you have to give anyway. Now I said can I go for another? Okay. Tabulating data, because tables are often used when you are writing manuscript. That's one of the commonest use a form of, apart from the, let's say, a text, that's a way of, illustrating data. And then I find most of the time that the tables are very badly done. There's a principle saying that data should be tabulated only when it is possible to present them in more concretely in that form than any other form. So if there are any other form that can be chosen, you must choose that, not a table. Now, if you look at a table like that, I don't know why you had to bother having a table. You can very easily write that saying that information in just one sentence. So you don't have to create tables. So sometimes you find the manuscript which are about having 25 tables ready, and each table has a very little information. So that can be put into just one sentence and forget about it. Okay. And also tables are not for the data storage, right? And absolutely this kind of tables should not be drawn. So they are not for the actual data storage. The tables readers find specific information on exact numbers, and the figures show comparison of trends. I'll give you an example of one of our publication, and this is what we showed about how the bone mineral density changes men and women for the period of time. So that's very well shown that the men are actually staying higher all the time compared to women, and then that's bone mineral density I'm talking about, and then how it behaves with the age. So you can see around the age of, let's say, 50, start declining. So that's very, very nicely graphically shown. But then if someone wants to know the exact numbers, we have given the exact numbers in a table as well. So you think that some people would like to know the numbers really, so I've given the numbers, and also we've given that in the graphic form. Now in the table, in a table, there are what are called the components. For example, there are what are called values. Values are the information is an individual cell. In that individual cell, I'll come to that, in a single datum, a symbol or an observation, you find it within a cell. And then you can see that the cells are lined up either as rows or as columns, and they are what are called group data. So group data are the cells which are commonality are grouped together in columns or in rows. And then there are what are called comparisons. That is the information given in the rows, so the columns are compared and given another column for that. Just to make it is easy for you to understand, this is in this taken for the NEJM, and this is of course what is called the cell. This is in a cell. You can see that a datum, right? Single data entry point. And then these are what are called the columns, right? And these are what are called rows. These are grouped data. And then you have got what is called comparison. Hazard ratio is a comparison between. So that's how you organize a table. Little more. So table has what is called, these are the what are called elements in a table. There's a table number. So if your manuscript has only just one table, don't say table number 1. Because it's sometimes, I find that, some people, there's just only one table or one figure, say figure 1. You number it only if there's more than one. If there's one, you don't have to. You say table. You say figure. Refer it as a figure or refer it as a table, not a number. It's not necessary. And then there's a title of the table. I will come to that later. 3 lines. There are 3 lines. I'll show you. And there are columns, rows, data field, and symbols, and the footnote. That doesn't make much sense to you unless you see this. Now it's a table. and the number, if required, and there's a title. So this what, and there I said there are 3 lines, right, for here, and there's one here, and this column 1, column 2, data, and there are row heads 1, and then there may be subheads. There are what I call the footnotes and the expanded abbreviation. So that's actually what are called elements of a table. So you can see that, when a table is constructed like that, this is how the table should look like. 3 lines. Don't put a lot of lines, vertical lines, and not get rid of all the vertical lines. And then there's what's called the using symbols. And then there should be some kind of thing. Let's say you want to say something about the column 1, you put an asterisk, and you want to say something about the explain the column 2, you put 2 asterisks here. And then you have to say something about the row head number 1, you put 3 asterisks, and go on like that, putting asterisks. So you'll end up with about 10 asterisks in this thing. Is that the right way to do that? This is the right way to do that. The footnote reference in a table. First you get asterisk. And then if you want mother, you got to go what's called dagger. And that's what's called double dagger. There's a section, double vertical line, and pilcrow. And you get repeated. You get 2 asterisks, 2 dagger, 2 double dagger, 2 sections. Like that, you get repeated. So this is what's called the most standard way of giving what are called the symbols. Not running into maybe 10 asterisks or something like that. Is it really happening? Yes. It's an NEJM. NEJM using that, you can see that in the footnote, they're using all that, symbols in that manner. So that's the right way of using symbols, not asterisks. If 1 or 2, you can certainly use that, but then if you're running to about 3 or 5, you should not do that. Organizing columns, what are called column heads and the cutting heads. This is what's called column head. Okay, and then the column 1, column 2, and then you can have what are called this is one table constructed in that manner, and then what are called cutting heads. Under columns, you can have small headings, so cutting heads, right? So likewise, so this is one of the table that we have constructed: percentage bone mineral density change, male and a female, and then type 1, type 2. This osteogenesis imperfecta, type 1, other types, type 1, other types, like that. Right? So it's a little more organized. Same way, there's a common head and there's a column head. The principle, again, table should have a clear purpose. So if you have a message, and make sure table, text or figure is not this, I already said, and you can spend some time to construct a table. And people think it's one of the easiest things to do. It's not the easiest thing to do, really. Now look at this particular table for a change. For example, let's say comparison of computer literacy between health professionals. This is a comparison between house officers, registrars, and the consultants. Okay, on their knowledge on what is MS Word, MS PowerPoint presentation, and also Excel. So just have a look at this one for a minute and see if I did that in this manner, whether that is more clear. Now you can see that when you see that, let's say instead of that, if you see that, you can see the consultants are doing really pathetic, isn't it? Okay? You can see that's more evident in that manner. But it was not evident in this one. But when you put it this way, it's more evident. Why is that? Because our brain is trained in such a way, this is the entry point of a table. You enter into this one and read this way. You always, your brain is trained in such a way that for a table, you can see that, you generally enter into this point and then you tend to read this way. That's how the human brain works. So because of that, you give the information in that order, the natural order. So if you gave that in the natural order, you can see that how the consultant, the 3 groups of health workers get compared with regards to their skills in the different components of Microsoft Word. Okay, which is much clearer. What is this? Yeah, this is another thing. This data presented in a table should not be duplicated in the text. This is one of the common error that people make. Let's say all this information given, this table in the first para that I have written here, all that is given in the table. And you write that, these numbers and then give a reference in the table 1. You should not do that. You can say the compared subjects in the control group, patients in the treatment group was older, heavier, and taller, and then give a table 1 as a reference. And then you know that if you want to find that, you have got the information here, and then if you want to find that in the more details, you can go to table number 1 and see that. So in summary, I'm sure taking more time. The methods and the research are the main pillars of the manuscript, and they are really holding the argument. But people don't read them really, because as Professor Roja Singh said, most of the people, they read only the title, and then some people will go and read the abstract, and then introduction a little bit, but not the method and the research generally, and then there's a discussion. So methods and results are generally not, in fact, there was in RAD format, and there was a suggestion that at some point, push the method and the research section beyond the discussion. There was some argument, some discussions at some point saying that it should be changed, and then bring the introduction, the discussion, and they push these things down so that people will, only the interested people will read. And the inadequacies of reporting, very common. It's actually inadequacies rather than the inadequacies in the methods that you have, your methods have gone through the ethics, and the ethics have sort of, the research committees and all that, they have looked at your methods and said your methods are fine. But it's the reporting quality is actually the problem. That's what I said, those guidelines are really useful for you to go through and see whether you are doing the right thing in the right way reporting. And they are the main reasons for rejections. And the method and the research should be well organized. I said they should be concord. There should be consistency between the two. And there are many guidelines and the checklists that people can use. All right? Thank you.
Thank you, sir, for the comprehensive and very interesting lecture. As I said earlier, if the research section is the engine of the manuscript, the wheels are going to be the discussion, which will take you from unknown to known. I think we have Professor Anuj Premavaradhan, who is going to talk to us on how to write discussion and conclusion. As you all know, he's a professor, senior professor in medicine, attached to Faculty of Medicine, University of Kelaniya. He's one of the top-rated, eminent research in the field of clinical medicine, particularly in the field of hemoglobinopathies. Over to you, Anuj.
Good morning. Thank you very much for giving this opportunity. Listening to Professor Saroj and Professor Lekam-Vasam, I understand yet again why my papers get rejected, even with such an advanced age. Mostly probably because of the results section, I think. And writing a paper is not an easy task at all. It is it's a skill set which you need to acquire. And I heard, I think Professor Lekam-Vasam telling that in the morning, trying to teach people to swim with distant education. It says something a bit like that. You got to get people to write and then learn how to do that. But it's a craft that you learn, and the more you write, the more you fail, the more you learn. And failure is definitely part of the game. And it's being creative, as I think just like writing a song, making a movie. And that creativity part of a paper is the ability to spin the story. So there is creativity in deciding the topic. That's really necessary. You have to decide on a very good area to do the research on. And then, of course, hopefully you get good results. But once you get the results, you need to know how to spin your story so that it's marketable. And that's where the discussion comes in. Now, it's it's important to know the theme that I was given by Metta was discussion, conclusions and references. I'm keeping the references out most of the time. Now, discussion and conclusions are not always there in a paper. Some papers would have only a discussion. Some papers would just call it a conclusion. Some, on the other hand, will have a discussion and a conclusion. For example, one of the premier hematology journals, Blood, has only a discussion. There's nothing called conclusion. So the conclusion has to be incorporated into the discussion. Whereas British Medical Journal has a discussion as well as a conclusion. So the lesson here is when you write, before you write, you have to learn, you have to know which journal you are targeting, and based on that you have to design your manuscript. Now this is again for those people who are used to the game. You know that if rejection is part of the game, so you might have to keep on changing the style based on what your result is after your first submission. So you have to be prepared for resubmissions, and if it is for a new journal, the way you redo might have to change. Overall, the discussion part, or let's say discussion and the conclusion, should not be more than a third of the entire paper. So in a very big paper like the manuscript or a review article, which is about 3,000 words, the discussion component should be maximum about 1,000. So that's something which you should bear in mind. Now, the discussion essentially carries the analysis of your results. Now, you are basically trying to explain why this happened. To some extent how, but mostly why something happened based on the results you generated. So the larger implications of your research is being told, and then of course the connotations of your results. So a successful discussion puts your findings into context, the results, the interpretation of the results. It's very, very important to realize you don't repeat the results, you interpret the results, then you put it in context with what is known, and then you try to look forward based on what you have found. That is in a sense what the discussion is. So you ask certain questions from yourself. So your entire paper would be based on a hypothesis. So you ask a question and your research is trying to answer the question. So in your discussion, you ask yourself, was your hypothesis correct? Maybe it was correct, maybe it was not, maybe it was only partially correct. So then you got to ask, try to answer, why was that? And that should be based on existing knowledge, based on other studies, conflicting results. Always incorporate results or other studies which are not in keeping with yours. So an analysis of other data which you have from other people. And with all of that, finally, make sure you give a take-home message. So a discussion can be, not necessarily always, be written in 3 paragraphs. The first paragraph is interpretation of your key findings. Again, not a repetition of your results. It is not a repetition of your results word for word. You interpret the results and try to explain what the results mean. And the second paragraph would be comparing and contrasting with previous studies, highlighting the strengths and weaknesses of your study. Not just the strengths, but also the weaknesses of your study. And this is the point where if you have an unexpected finding, you can introduce that. And the last paragraph is to summarize the hypothesis and the purpose of the study and highlight the significance of the study and give a take-home message and suggest a future course of action based on your research. So generally, journals suggest certain do's and do nots for the discussion, and here are some of them. First and most importantly, read the journal's guideline and have a very clear idea about the word count, the style of writing, and you start the discussion with a statement of the principal findings, not the introductory statement. So, for example, if you are doing a work on, let's say, SGLT2 inhibitors in diabetes, and to see whether it causes liver disease. You know, I'm breaking new ground here. You don't start by saying you did the study on SGLT2 inhibitors to look for evidence of liver disease. That's not the place to do that. Here you start the discussion by saying, in our study, 56%, whatever, had evidence. So you are analyzing the data and giving the information and immediately start interpreting that. So this is not the place for an introductory statement of the topic. So you don't introduce it again at this point. You start with the results. So you get on with the results. And you have to state whether the results prove or disprove the hypothesis. So during the process where you give the data, so your story is about SGLT2 inhibitors and whether they cause liver disease or not. So you go on to talk about how your data proves or disproves that, and go on to find ways where the research can progress. But again, learn to be concise. This is very important. Do nots include rewriting the abstract, including new arguments. Now, this is very important. You have the ability to use references during the discussion. You can introduce new references during the discussion, but they should be within the scope of a concept which you have already discussed. You can't introduce a new concept for the very first time. So something which is completely alien cannot be introduced for the very first time in the discussion. So this is very important for you to understand. If there is a discussion and a conclusion, you can never use a reference in the conclusion part, whereas you can use a reference in the discussion part. But you can't introduce a new concept in the discussion. But you can use references about something which you have been using in the introduction, which is well within what you have been writing. I'll give you an example about this. And never apologize. Don't berate yourself for not having this and not having done that. But that does not mean talking about your limitations. You should discuss your limitations and your negative results, but do not be apologetic. You should discuss this in a scientific way. And as Professor Lekam Masam mentioned, don't overstate, don't overglow about your positive findings. Don't try to make a big show about what you have found. Just specifically mention about what you have found. Don't make grandiose statements about what you think you may have found. So, yet again, effective discussion would be a place where you summarize the key findings in a clear and a concise language, acknowledge the hypothesis if it is not correct, place your study within the context of previous studies, discuss the potential for future research, but provide the reader with a takeaway statement at the end of the manuscript. Now, most of you PGs are very familiar, I'm sure, with the Emparage outcome study, 2015. Now, in that particular, I'm using this to highlight how they have used their discussion and to show you that you really don't have to stick to a very strict guideline. You know, people have the liberty to write in any way they want, provided that it makes sense. Now, I'm sure for most of you, facts are more interesting than about writing styles. For those of you who are familiar with Emparage, this is a study where they looked at the effects of empagliflozin. There were two arms, or actually 3 arms, 10 milligrams, 25 milligrams of empagliflozin versus standard care, to see whether there was increase of cardiovascular mortality and all-cause mortality. Right? You remember this. And a total of 7,020 patients were treated for a short time, 3.1 years. And there were 2 outcomes, the primary outcome and the secondary outcome. The primary outcome was all-cause mortality, deaths or ischemic heart disease or myocardial infarction, strokes, non-fatal strokes. And the secondary outcome was unstable angina. So what the study found was that the primary outcome was significantly reduced with empagliflozin. It didn't matter very much whether it was 10 milligrams or 25 milligrams. But the secondary outcome, unstable angina, did not reduce. And it was, they pooled the data of 10 milligrams, 25 milligrams versus the placebo arm. And so in this paper, in the New England Journal of Medicine, in the abstract, there was no discussion, it was just a conclusion. So the conclusion was patients with type 2 diabetes at high risk for cardiovascular events who received EMPA as compared with placebo had a lower rate of primary composite cardiovascular outcome and of death from any cause when the study drug was added to the standard care. Now, this is how they discussed this. Now, it was a very long discussion. Now, they didn't really stick to, the 1,000-word. This is the New England Journal of Medicine, and this is a huge paper. And this went on and on. And I color-coded it. So the first part is where I would have included the analysis of the results. The green is to say, to contrast with other data. It really doesn't actually. It is not taking into consideration any other studies. It is going on with its own analysis. It's too big a study, I suppose, to look at anything else. And the final, of course, it has a take-home message. But for academic purposes, let me just read this to you. So the discussion goes like, among patients with type 2 diabetes at high risk of cardiovascular events, those receiving empagliflozin had a lower rate of primary composite outcome death and cardiovascular causes, non-fatal myocardial infarction, non-fatal stroke than receiving placebo. The difference between empagliflozin and placebo was driven by a significant reduction in death from cardiovascular causes, with no significant difference between the risk of myocardial infarction and stroke. So they are analyzing. They are not just giving the numbers here. They are analyzing, saying why the deaths were not different. They are not giving the numbers here. Since the two groups had similar rates of hospitalization for unstable angina, the secondary outcome, there was no significant difference in the key secondary outcome, which included the risk of hospitalization for unstable angina. Patients in the empagliflozin group had significantly lower risks of death from any cause and for hospitalization for heart failure than did those with the placebo. Although a small dose response effect for the 10 milligram of empagliflozin versus placebo and 25 milligram versus placebo has been documented for metabolic response, in our study, the two dose groups had similar hazard ratios for cardiovascular outcomes. Thus, in clinical practice, the choice of empagliflozin dose will probably depend primarily on the achievement of metabolic targets. So they are going beyond the scope. So they are talking about the differences between 10 milligram and 25 milligram and talking about saying they are being critical, that it's not, doesn't seem that 10 and 25 is going to have a huge difference on the heart. It seems like more likely to be metabolic. So analysis is going on. These benefits were observed in the population with established cardiovascular disease. Now this is important, in whom cardiovascular risk factors, including blood pressure and dyslipidemia, were well treated with the use of renin-angiotensin-aldosterone system inhibitors, statins, and acetylcysteine aspirin. The reduction in the risk of cardiovascular death in the empagliflozin group were consistent across the subgroups. In other words, in this passage, they are talking that these results may not be applicable into other people who are not being as well treated with all these drugs. So here they are analyzing the results and saying they are comparing and contrasting not with other studies, but empirically. They are saying don't interpret these results to a group which is not well treated with other drugs. So discussion is an analysis of their results and giving wisdom to the reader. So be careful when you're interpreting this. And then here is an example where they are using references. This is very important for those who are writing, that you can use references even in the discussion part. They're talking about the effects. This is an interesting area. Here we say, these guys say, our trial was designed to assess the specific effects of empagliflozin on clinical outcomes. And the mechanisms behind the observed benefits are speculative. We are not going to talk about the mechanisms. As such, we infer the mechanisms behind the cardiovascular benefits. The trial was not meant to argue out why it happened. We just looked at whether it happened or not. But in case you are interested, we think the mechanisms might be, and they are giving references. And goes on and then concludes by saying, in patients with type 2 diabetes at high risk of cardiovascular events who received empagliflozin had significantly lower rates of primary composite cardiovascular outcome and death from any cause than did those in placebo group when the study was added to standard care. So there's a final crunch take-home message. So it's the part they are able to sell something, but being critical as well. So if you have a chance of writing a conclusion in addition to a discussion in a paper, which as I mentioned is not always the case, then in that situation, there you have to restate the topic. This is a study of hemolytic anemia in 20-year-olds in Bahamas or something like that. Restate the thesis that you presented. You are looking for the prevalence of gallstones in Bahamians with hemolytic anemia or something like that, and briefly summarize your points and then call for action. So it has a completely different objective than the discussion, where you are, whereas the discussion is a point for analysis, the conclusion is revisiting the entire topic, reminding the reader, this is why we did this is what we found, so, and then giving a take-home message. And yet to bore you with a little bit about diabetes again, this is a meta-analysis, benefits and harms of drug treatment for type 2 diabetes, a systematic review and network meta-analysis of randomized control trials, very, very recent, 2023 BMJ, which compared 13 drugs for diabetes, looked at benefits and harm. And this is the conclusion. And again, color-coded, restating the topic, restating the thesis, summarizing the points, and calling for action. So what it has done, when it re-looked at multiple treatment options, so what it did was, keeping pace with the growing number of published randomized trials in adults with type 2 diabetes, this meta-analysis finds highly heterogeneous benefits and drug-specific harms across 13 drug classes. And so in blue, it summarizes what it wanted to say. The main key findings are, beyond confirming the substantial cardiovascular kidney benefits of SGLT2 inhibitors and GLP-1 receptor agonists, with absolute effects highly dependent on the patient's risk profiles, we find that finerenone and the drug recently made available displays quite similar benefits to SGLT2 inhibitors and GLP-1 receptor agonists. Tirzepatide shows superior benefits on weight loss and SGL2 inhibitors and GLP receptor agonists. So it argues out about these two new drug groups, and the final take-home message is these results and other key findings of our comprehensive systematic analysis highlights the need for continuous assessment of scientific progress to introduce cutting-edge updates in clinical practice guidelines for people with type 2 diabetes. So don't stop. Just continue to look at the updates, because there are new drug classes coming in. Otherwise, you go back. That's essentially what it tries to say. So if you do have a chance to write a conclusion, again, restate the problem, summarize the overall arguments, and suggest a take-away message from your paper. Okay? Now, the last part, which I'll be touching very, very briefly, is what do you do about referencing when you're doing a paper? Just few tips only. While you write, now this is about manuscript writing, not about writing a thesis, a PhD, where you will have to collect about maybe 4,000 references. So this will be about maximum about 30 or 40 references maximum, hopefully. Keep on collecting the references. Don't wait till the end and you will forget what the references are. It doesn't matter what style the reference is at the time you collect it. Make sure you collect it in whatever, not necessarily in a reference manager, just whatever. Don't let yourself forget what the reference is. Indicate in the text where you are going to put that reference while you keep on writing. Okay, put a number, whatever. And when you're using a reference, always read the reference. Don't just put a number and a name and think what it says is correct. Read the reference and make sure that the reference is actually talking about what you think it says. And the references come in two forms, the Vancouver style and the Harvard style. For most of us in scientific writing, what we use is the Vancouver style, which is the numbering. So when you are using a text, put the number. So it is 15, and at the end of the text, you will put 15 will be whatever. Okay, so use the Vancouver style. Harvard style is mostly for literature, art and things like what Prof. Saroj is more interested in these days, I think. And so this is Harvard style, where you put the name, Das 1915, like that. So for us, our style, for most of the medical writing, we use the Vancouver style. And so it is important to first identify the appropriate source and the citation style, which is very different from what I am saying by Vancouver or Harvard, could be APA, MLA, Chicago. That would depend on the submission paper. So whatever paper you are going to submit it would specify which format it has to take. And if you are going to be a regular writer, which hopefully you would be, start using a reference manager. And when you are, again, depending on the kind of manuscript you write, whether it is a full-length manuscript, whether it is a case report, the number of references you are allowed would be limited or specified. So again, be aware how many references you are allowed. So be aware about that. And remember, the style of referencing would be different from a journal when you are taking from a journal, when you are indexing from a textbook or from a website. Now these are very easy to find out. And this is just to say, for example, you are referring from blood. It has a very specific style. They would say, use the blood style. So it is not using Vancouver, it is not using APA, it is not using Chicago. It has a blood style. So you have to use that particular style for that particular journal. So this, again, use instructions, essentially read instructions to authors before you start writing and submitting. The trial run, which I was trying to do, I think I'll just pain Nanny. I can't. I don't think I can show this to you. I can give a trial run for you to write a paper if you are excited enough to do that. Just give it a try. Maybe one of my registrars, so hopefully she is listening, had a huge problem in writing this case report. So I'm trying to give it to you as a challenge to see whether you could make any sense of this. This is, I'm not sure whether I'll be able to give you the full data on this presentation. We recently came across 2 thalassemia major patients. This is the background to this. Okay, so what your mission is to write a case report, the discussion part of this. Whoever writes it well might get a publication. That's the These 2 thalassemia major patients have hypertension, systemic hypertension. So the exciting thing about it is that systemic hypertension is not known in thalassemia major patients. Never published. No patients ever in the literature, in clinical practice, has never been come across. So there are 2 patients. And so these patients were investigated for secondary causes is being investigated right now. And so a case report, usually maximum word count is 1500. You can't exceed that, the maximum full thing. Is anyone interested enough to try it out now? Anybody? Can give the data if you are up for it, otherwise I'll close shop. Would a publication excite you? If you're interested, maybe you can contact later. Maybe. So anyway, I'll give you the data. You can. It's just so that your mind starts to work. So this is the introduction we wrote. Okay, the paper is not written yet. So it's not final. Okay, this is just something which some of us wrote. So cardiovascular complications, this is the introduction, it's not the discussion. Cardiovascular complications are a significant cause of morbidity and mortality in transfusion-dependent thalassemia. Iron overload related cardiac disease, pulmonary hypertension, not systemic hypertension, are the two commonest cardiac abnormalities to affect them. Myocardial infarction, heart attacks, too has been reported quite infrequently. There are only 3 reported cases worldwide, MIs. Systemic hypertension is not encountered in thalassemia patients in clinical practice, nor are there any published cases on this regard. There is evidence to suggest that iron overload may cause increased medial thickness and increased atherosclerosis, but no corroboration with systemic hypertension has been recorded to date. So, you go on to say, we observed 2 adult patients with thalassemia major from the same unit. These are from Kurunegal actually, with sustained increase of blood pressure, and decided to investigate for a possible underlying cause. The attendees of Kurunegal, which looks after about 850 patients with thalassemia, of which over 500 are transfusion-dependent beta thalassemia. So puts it in context with the number of patients there. And so the case histories are this. Patient 1, 26-year-old, thalassemia major, diagnosed at the age of 6 months, been on regular transfusion, monthly transfusions previously, now every 2 weeks, received over 356 transfusions lifelong, mean pre-transfusion hemoglobin over the last 12 months 8 grams, should be around 9, now it's 8. The last examination, her spleen was very small. She was started on chelation at the age of 2 years, subcut desferrioxamine, and at present using combination. The current serum ferritin is very high, 4,000. Highest ever ferritin is 10,000, very high. She attained menarche at the age of 15, induced puberty with low dose estrogen. Now she gets her periods regularly unassisted. She was diagnosed with diabetes at the age of 14 years and at present treated with premixed insulin, 30 units and 20 units. She had a last ophthalmological assessment 2022 and no neuropathy. Hypertension was first noted at the age of 26 years ago during a routine pre-transfusion testing, and at present she is on captopril and bisoprolol. There is no family history of diabetes, hypertension, hyperlipidemia on any of her first degree relatives. Patient 2, 30-year-old, thalassemia major. She's, I think, thalassemia intermediate because diagnosed at 3.5 years. Regular transfusions. Transfusion load, same, very high, 354. 7.5 HP, spleen very small. Chelation history is there, ferritin 1000, highest ferritin 5000. Menarche is spontaneous, but at 16, so delayed puberty. Diabetes at the age of 14, now she's 30 years, treated with premixed insulin. Last eye assessment has no retinopathy, no neuropathy, hypertension diagnosed 2 months ago, no family history. So that's the background. And then this is what you might not actually see. Essentially, almost all the investigations are normal. So, except there is one which we are investigating in one lady, overnight dexamethasone suppression is high, 158, but low dose dexamethasone, it was suppressed. So anyway, so if you, in a scenario like this, how would you start writing the discussion? So it's not going to happen instantaneously, so I'm not going to waste your time on this. So what I try to do, just to show you, how modern technology works, I got AI to write an introduction for me, not a discussion. And this is Site AI, which wrote the discussion, which is writing rubbish actually. And so this is just to show, these days giving a talk on scientific writing without talking about use of AI is archaic. So we can use AI for writing, which is allowed. What we don't know is how to, as Saroj said, how to give credit to AI. And I'm very amateurish, and my technology is like Amasam said, I would be in that 55% who doesn't know how to use computer skills very much. But when I got AI to do the writing, I found it was the text it was giving and the references it was giving is junk. So you have to be a bit careful when you're using AI. Don't believe everything it says. For example, in the introduction it says, male gender has been associated with a high risk of cardiac iron accumulation and fibrosis, heart dysfunction and complications in thalassemia major patients. And then came this other reference which says patients with beta thalassemia at a high risk of developing systemic hypertension, which is not so. There's nothing, there's reference 7, right? So when you start using AI for writing, there are so many sources, Jenny, ChatGPT, and there are I think about 15 sources of AI now available which can write, design PowerPoint also. You have to be very, very careful not to believe everything it says, at least from very amateurish point of view. That is what I know. Scientists may disagree. So anyway, this I'm not going to bother you with the rest of the paper, but so how would you just think, how would you formulate a discussion for a case scenario like that? My registrars are grappling with it right now, and anyway, I'll stop here. Thank you.
Thank you, Anuja, for the excellent talk on discussion and conclusion sections. I think we have come to the Q&A sessions. May I ask all three speakers to come to the table over there? Can we have one more chair, please? And those who are joining us here physically can ask questions. You can use a mic. Can we get a mic? And also those who are joining virtually can use a Q&A section or area of the Zoom platform. You can text your message. You can use that area to text your message, and then we will ask that question from the resource persons. A few housekeeping announcement. We are going to give e-certificate for all of you. So therefore, please make sure that you provide your correct email address. In near future, you will get the e-certificate from Sri Lanka College of Internal Medicine. And those who are joining virtually as well as physically, please provide your correct email address to us. And also, those who are joined virtually as well as physically, you will get an email from the college on feedback, Google form link. Please fill that feedback. That will help us to improve our workshops in future as well. And the refreshment will be provided at the end of the workshop outside for those who are joined physically only. Though. Right, if there's any questions, you can ask now. In fact, I have one question from that I want to ask from Professor Saroj Jaisingh. In fact, you said that the keywords are there mainly for the purpose of searching the paper. I'm thinking whether, because I have noticed that sometimes the same keywords are used for the title also. Whether it's going to duplicate the thing, because the title also we use as the for the searching purpose, and then of course we have the keywords. If you use the keywords which are there in the title, don't you think that's duplication, or whether we are allowed to do that?
I think we can do it. It's okay. There's no hard and fast rule. But I think if you have the keywords also in your title, right, what you mean is if you use other words, you might
No,
capture...
I'm asking the same keywords. For example, say prevalence of diabetes in Sri Lanka. Probably you can use, say, prevalence of diabetes as a keyword. Then, of course, that is there in the title also.
That's okay, I think. No? What do you think? What about the others? Anuja?
That's not a problem,
Yeah, then what is the purpose of using the prevalence as a keyword? Because the prevalence is there in the title also. Then of course you are using the prevalence as a keyword also. Whether there is any added benefit to using the prevalence as a keyword in there. Because prevalence is there in the title also. And if you use the prevalence as a keyword, sir, because there is no added benefit, I suppose. So there is argument whether we can use some other word which would improve the visibility, because then we have a higher number of words as the search, when we search the paper, whether that would improve the visibility.
Yeah, I think you've got a point. I suppose it depends on the search engine we are going to use, because if you are using Google, I suppose what you say becomes important. But if you are using a database like PubMed, I think the keywords corresponding to the title or the abstract would be useful. What do you think?
It depends on, let's say now, in the PubMed there are two types of searches. No? There's one called, what is called free text. In the free text search, whatever the word they are in your title, your affiliations, or in the text would be picked up. That's called free text search. But then there's what's called mesh. Keywords are important mainly in the mesh search. Because when you have, let's say, a list of keywords, and which are alphabetically arranged, and then the staff in the National Library of Health, that's right, yeah. They use those keywords and they modify them, and then use those to link your paper into various places in the mesh tree. Now there's a mesh tree, and the mesh tree goes various directions, so your paper get attached to many places in the mesh tree. For example, let's say if it's randomized clinical trials, it goes as clinical trial, randomized. That's how they put it there in the mesh people. So there it will get attached into under randomized clinical trials, randomized. Let's say it is under, let's say, one of the drug, and then the particular drug name, it is get attached into that place as well. So then your paper will be attached to a mesh tree in different places, right? And using your keywords. So that is mainly for the mesh search. But again, if you are using the pretext search, then which most of the people are doing that, you just punch your word in that box and then search, and then there is a pretext search. And then if that particular word that you are using for the search is there anywhere in the article, and that will come. So there's no problem.
Yeah, I think I was just checking actually, because I think this relates to Google, your search engine optimization and the words you use. Because what you get with Google would be different to what you get with PubMed.
Yes.
Right? So, and now with ChatGPT, you can get another set of they will also come up with their list. Yeah.
Any question from the physical audience?
Thank you very much to all three speakers for the excellent talks. I think I learned a lot by being here today. Just a question to Prof. Anuja. Now, listening to you, I gathered that there are different styles of writing, the discussion and conclusion. So is it that you would increase your chance of acceptance to a certain journal if you study the discussion style or the conclusions that they tend to favor, or is that something that you can disregard and write, if I may use the word brutally scientific, and write an analytical discussion?
It's a two-way thing. That is, I think, if you want to aim high on, getting it published in a good journal, I think you first want to see the style they want, and but a sense of the story is going to be the same. But what the author guidelines would tell you would not be the style of writing. It will give you the basic essence of the word count and things like that, whether they will allow you a discussion and a conclusion. But looking at the previous articles of that journal will help you to see the chances of getting published in a journal like that based on the results you have. Your style of writing is going to be your style of writing. I don't think that's going to change very much. Essentially look at, before you start writing, first decide on where you are going to pitch. First pitch as high as possible, not as low as possible. For people who are starting for the first time, pitch for the highest possible. Obviously, there is a very high chance of rejection. That's fine. Name of the game is to learn from it. Don't get disheartened. But if you are not rejected by the editor, in which case you will be, rebuffed on the very next day itself, then you will learn from it, because there will be some critical comments, and that will help you to maybe not to resubmit to the same people, but at least to someone else. Editors, writers' guidelines would help you to structure it, but the style of writing is something you learn on your own, I suppose. So, isn't it? So,
I think the other important point is which journal. When you mentioned about the journal, these journals now give a way of finding out by, can search the journal, which one will prefer your paper. For example, Elsevier will give, they have their journal search. When you go to that website, you type in the title and the abstract, and they'll give you suggestions for journals. So that's something which helps. Now, the second point is, don't be disheartened by rejection. I can still remember the first paper I went through was 8 times, and that was a time when we had to type with a typewriter each version. Now you have the luxury of computers doing it. Each version had to be typed. Even currently, there are 3 papers of mine in the queue, rejected 4 or 5 times. When the first rejection comes from the editor, as Anuja says, you don't learn much from it, but you get knocked out. But if it goes to the reviewer and gets rejected, then that's useful, because the reviewer will give you some ideas and pick holes, and then you submit to the next journal. And so as you, free of charge in a sense. But so you progressively improve, and you can hit a very good journal. I mean, there was one paper, which of course it was a concept paper, but which came up in Mayo Clinics. I had about 5 rejections before, but then suddenly you get success. So don't be disheartened by the journals. And the third point is about predatory journals. You must be very careful, because there are lots of journals. The moment you submit, the hackers know that you have submitted, and you start getting invitations to publish in journals. Invariably, when you look at those journal sites, you know that they are predatory because they look very odd, right? And the titles of the journals are also a bit odd. And the people who write to you are invariably it's .coms. It's not .org. And there are a few clues like that. And invariably, once you submit, after some time, they will ask for fees. They say rapid publication, and they say $50 or something like that. So you have to be very, very careful. The Beals list is it's now continuing, I think, because you can find whether it's a predatory journal or not. So the safest is to go to the PubMed or Scopus or one of those well-recognized databases, and the SCI. Then you know that those are recognized journals. And as Anuja said, you start with the reasonably high and impact factor, and then you can come down after that. So that's how I generally do it, but I don't know. What do you all think? Yeah.
I think Satish's question was that the discussion should be written in such in a manner that which enhances the chances of publication. I have really what I've seen is that when you get when a paper gets rejected, and there are some instances. For example, when you see a paper comes from let's say developing country like Sri Lanka, some editors or the reviewers just do not take into consideration, and that is a very unfair kind of situation. But a fair reviewer would look at mainly the methods and the results section, and then because he or she knows that the interaction and the discussion part can be rehabilitated, but not the methods and the results. So if the method and the result sections are sound, then they will be very happy to sort of suggest amendments to their introduction and say that your introduction is not focused and not taken into consideration, let's say, a small body of evidence in the local literature or something like that. They will be give a very specific instruction how you can sort of edit your introduction and the discussion part. But generally, I haven't seen that paper get rejected simply because the discussion wasn't right, because that can be amended. So there's no specific way of writing and all. And that's what I feel about the discussion part.
There's a question from one of the virtual audience. I think to Anuja. Should the discussion section also mention probable deficiencies in the methodology used?
Yes. If there are things, like for example, the answer is definitely yes. If you found things which you yourself know, for example, sample size, you could have improved on the sample size. Yes, so when I said don't be apologetic, I'm on the next thing which I said is that you should be self-critical. So you talk about for example, it could have been different if we had a larger number of controls, a larger number of patients, if we had recruited people from multiple centers. So you're being self-critical there. The answer is yes, of course. I think the methodology, as Prof says, methodology is the key. So if there are if there are major methodological errors, you being self-critical is not going to make the paper any better. It will be rejected outright. But if you can explain and suggest ways to improve the results, I think that would be accepted.
Can I make two other points if there time? One point is that about when you start submitting, they will ask you all sorts of questions about conflicts of interest. Now, be honest and try to fill those. And conflicts of interest does not mean that you get ruled out from a paper. It's just that you have to express it in the submission. So that's something you have to be aware of. And also the other point is, of course, I am a supporter of case reports. I might take up that challenge because I think that's what clinicians excel in. In a sense, more than research, research is, I think, is required for us to understand the advances and to critically appraise the literature and to incorporate it into our clinical practice. But as clinicians, the key would be the case reports and case studies, because your clinical reasoning is the crucial. You must know what is going on in your patient before you apply all the knowledge. There will be volumes and volumes of guidelines and textbooks which will tell you what to do once the diagnosis is made, right? It's only after that that you know what to do. So if you don't get that, if the clinician doesn't get that fact, that step correct, everything else is bunkum, because if you haven't got the diagnosis, you don't know what to do with that patient. Your investigations, treatment, everything goes out of the window. So that's where clinical reasoning and case reports come in. And I would actually urge the college to have another session on these case reports, because that's our bread and butter as clinicians. Yeah. Thank you.
Thank you, sir.
There's some more questions coming.
Right. I have no access. Can you, sir, if you are?
If you could not find the expected sample size within the expected time, example in cohort study, lost to follow up about 40%, can we still publish what we found? You want me to answer that?
Yes.
There are actually two things that in this question. One is that you are not meeting the expected sample size. The second thing actually what is posing is that what is called loss to follow up. Those are two things really. For example, let's say you thought that you need a sample size of 200, but then within the time frame and also with the resources that you have provided, and then let's say prices went up and all, and then you could manage only, let's say, 150 people. That is, you had let's say 50 people less than you expected in that. Now, there's no way that you can sort of, let's say, recruit more people and also. So you should not get disheartened. What you should do is actually sample size calculation, what's called the priori. That is, before you start the study, you do a calculation, sample size calculation, that's called priori, before the study. And take into consideration the estimates or the experience that you know of the research of the other people. So let's say if you are looking at the prevalence of, let's say, smoking, whatever it is, that using the prevalence that other people have found, you calculate sample size. But now, at this point, you have a luxury of, let's say, looking at your data. Now you have got what is called the prevalence that you have found in your study. So now you can do what is called the power calculation, post hoc power calculation. It's called post hoc, after the study collection of data, post hoc power calculation. That is in contrast to prior sample size calculation. So post hoc calculation will tell you that you know the sample that you have got, you know the estimates now for size, and you can calculate what the power of your study. Your power of your study, keep at 80%, 0.8 at the beginning. Now you can say, now with the observation that I have made, I have this study with 150 sample, gives a, let's say, 85% power. Study has 85% power to detect a reject a null hypothesis. So you can very well, you can publish it. There's no question about it. Okay? So even if you've fallen short of your sample size, there's a chance of you to publish it if your study has a sufficient power to detect that difference. So that's one thing. The second thing is what's called missing data. Missing data is a huge problem. If the missing is happening at random, okay, that is, I'll explain that. For example, there's a way that you can missing data in the random or in a systematic way. For example, let's say if you are doing a study on the something intervention on weight loss, okay, and you call the people to come, and maybe that the people who did not lose weight shy to come for the study repeat. So only the people who lost weight came for the follow-up, not the people who sort of, did not lose. So that is what's called systematic missing data, a systematic way. So that's not good. But maybe that, let's say you did a study, people could not take leave or something like that, come, and then there's a random of missing data due to random. And then that can be corrected, because there's something called imputation of data. So you tell your software saying that I have lost this much, and then I need to impute those numbers. So there are various ways of doing that, but if you meet a statistician, they will tell you. So the essential components for imputation of the data, so that is you put in data for those missing data, there should be two things. One is that the data, loss of data should be random, not systematic. Okay. The second thing is that it should be less than 10%. That is a little ballpark figure, less than 10%. As long as you have got a less than 10% missing data, and you have got it's lost in random, due to some random reason, and then you can impute it, and then you can create database. So there's no problem with that. I hope it answers that question.
As there are no more questions, we'll close the Q&A sessions now. I would like to invite Dr. Kohli Jayasundar to give a vote of thanks.
Good afternoon. Thank you very much. Thank you very much, Prof. Hera. It is a great pleasure to present a vote of thanks for the conducting interactive workshop on research manuscript writing conducted by our college. And firstly, let me thank this event coordinator for many years, Prof. Hera, for smooth coordinating and moderating of this event. And then I would like to thank our resource persons of this program, Prof. Saroj Jayasinghe and Prof. Sarath Lekamasan and Prof. Anuja Pemawardana is for agreeing this part of this program and conducted in super way and with their good experience. Then I would like to thank physical and virtual participants of this event, and I hope they had good experience with these eminent speakers. Then I would like to thank Dr. Kumudini Jayasinghe, the President of SLCIM, and the Council of SLCIM 2023 for giving the support for this event. Then I would like to thank for our sponsor, CCL, and Mr. Nalina for giving so much support for success of this event. With that note, let me finish. Wish you a very pleasant evening. Thank you.